Welcome to the Longevity Papers podcast.
Great to be here.
We're diving into the latest stack of research that landed between September 15th and the 19th, 2025. And our mission today, really tailored for you, our listeners in biotech research, is to cut through the noise.
Right.
We need that critical filter, a really high-fidelity look at these sources. We're not just here to, applaud every small step.
No, definitely not.
We want to pinpoint where the real novelty might be, look hard at the methodological weaknesses, and importantly, what are the hurdles to actually translating any promising findings?
And let's just set the stage right away. This past week, it's pretty much a perfect example of what we often see maybe, 95% of the time.
Yeah, sounds about right.
We didn't get that massive game-changing paper we're all secretly hoping for, the one that screams, add a decade to human life now.
Not this week, huh?
Not this week. What we have instead is good, solid, incremental science. We're seeing confirmations of pathways we already know quite a bit about, some really interesting new descriptive data, definitely, and a few correlations that, frankly, need some serious experimental poking before anyone should think about them as drug targets.
Okay, so our job today isn't just summarizing abstracts. It's about putting these findings under pressure, asking, is this really new? Or is it just a maybe more elegant confirmation of something we basically already knew?
Exactly. And if it does look novel, where's the gap? What's standing between this bench finding and a viable therapy people could actually use?
It's that distinction, isn't it, between getting a slightly better microscope versus inventing a whole new telescope.
Precisely. Biotech needs those paradigm shifts. Not just clearer pictures of processes we understand the fundamentals of. We need to know which targets aren't just going to, regress to the mean when tested properly.
So our roadmap today hits several key hallmarks of aging. We'll start with somatic mutation, looking at this dream complex paper that's got some buzz. Then we'll shift to chromatin stability, but from the nucleosome angle. After that, we tackle this big idea of epigenetic entropy and its supposed limit on lifespan.
Ah, yes, the 220-year question.
Exactly. Then we ground ourselves in human data with metabolic signatures from those amazing superagers in the New England Centenarian study.
A valuable cohort, for sure.
And finally, we'll look at a computational methods paper using AI to build better aging clocks, all sourced heavily, as usual, from the preprint server, the bioRxiv.
Yep, bioRxiv delivers again. Let's get into it.
All right, let's kick off with mechanism 1, somatic mutation and DNA repair. The paper getting a lot of attention is The Dream Complex Links Somatic Mutation, Lifespan, and Disease. This is Koch, Z et al., out of UC San Diego, posted on the bioRxiv September 18th.
Yeah, this one definitely caught my eye, and I think the reason is it targets what looks like a master regulator. It tries to connect the dots between keeping our genome stable and how long we live, even across different species.
So the Dream Complex, remind us DP, RB-like, E2F, and MUVB. Primarily known for cell cycle stuff, right?
That's its day job, absolutely, cell cycle control. But this study zooms in on its other role, acting as a central repressor for a whole suite of DNA repair pathway genes.
Ah, okay. So the hypothesis isn't just about controlling cell division day to day. It's arguing that this repression level actually shapes the long-term buildup of somatic mutations.
Exactly. That systemic burden of mutations, which they then link directly to lifespan, it's hitting that genomic instability hallmark head-on.
How did they build their case? What's the data look like?
Well, they put together a pretty convincing chain, starting small. They did single-cell atlas profiling across 21 different mouse tissues. That was key.
Okay.
And they showed that specific cellular niches, the microenvironments within those tissues where dream activity was naturally lower, those niches consistently had lower rates of somatic mutations accumulating.
So a direct link at the cellular level. Less repression, better repair, fewer mutations.
Seems to be the suggestion. Then they scaled it up, quite dramatically, actually. They did this cross-species correlation, looked at dream activity versus the maximum recorded lifespan for 92 different mammals.
92, wow. That's a broad comparison. And the result?
A pretty strong association. The trend was clear. Species with lower dream activity tended to be the longer-lived ones. It sort of hints at an evolutionary push towards dialing down this repression in species that stick around longer.
Interesting. Okay, so mouse cells, cross-species correlation. Did they connect it to human disease?
They tried to bridge that gap, yeah. They looked at clinical data focusing on Alzheimer's disease, and what they found was that lower dream activity in 80 patients seemed to correlate with a later age of disease onset and a lower risk of severe neuropathology.
implying that better genomic stability might delay complex age-related diseases like Alzheimer's.
That's the implication they're drawing. And then finally, the part that really gets drug discovery folks interested, the in vivo
By the
data.
intervention, what do they do?
They use a dream knockout model, specifically in mouse brains, and showed it actually protected against mutation accumulation over time.
Did they quantify that projection?
They did. They broke it down. For single base substitutions, SBS, your basic point mutations, they saw a reduction of 4.2%.
4.2%.
But for insertion deletions in models, which are often more disruptive structurally, the reduction was much bigger, 19.6%.
Okay, nearly 20% reduction in indels. That sounds more significant biologically, right? Indels can cause frame shifts and mess things up badly.
Absolutely. A 19.6% drop in indels is potentially quite meaningful, but, and this is a big but, this is exactly where we need to slam on the brakes and apply that biotech skepticism.
All right, let's unpack the skepticism. You said this was maybe the most interesting paper, but that 4.2% number for point mutations, it does sound kind of small.
It is underwhelming, frankly, when you think about the sheer amount of mutational noise that accumulates over a human lifespan, decades long. A 4.2% dent in point mutations, it's likely to get completely lost in the background static.
Just swamped by everything else happening over 70, 80 years.
Probably, especially in tissues that either divide slowly, like neurons, or turn over really rapidly, like the gut lining. Plus, you have to remember, DNA repair isn't simple. It's this incredibly complex network with lots of redundancy.
Right.
So you modulate one central repressor, DEAM. Does the system just compensate? Does it rely on backup mechanisms that are already working pretty well? Very likely. That 4.2% might be statistically significant in their experiment, but translate to meaningful human lifespan extension. That's a huge leap.
Okay, point taken on the 4.2%. What about the cross-species correlation? 92 mammals. Looks impressive on a graph.
Looks great, visually appealing for sure, but it's the absolute classic correlation versus causation problem, isn't it? Is low dream activity causing these animals to live longer? Or is it just a feature, a consequence of being a long-lived species that has already evolved a whole bunch of other superior stability mechanisms? We just can't tell from this data.
You can't separate association from cause just by plotting points.
Not at all. To even begin to make that leap, you need intervention data. Perspective, long-term studies, which points directly to the experiments that are missing, the ones the biotech community needs, before pouring serious money into targeting DREAM.
So what are those key translational hurdles? What needs to happen next?
First, the big one, safety. DREAM isn't just about DNA repair. It's tangled up with E2F and cell cycle control. If you systemically inhibit or mess with DREAM, could you accidentally take the brakes off cell division?
The cancer risk.
Exactly. That's a massive potential liability. You'd need incredible specificity, maybe some kind of allosteric modulator that only affects the DNA repair repression function without touching the cell cycle parts. That's a tall order pharmacologically.
So not just a simple inhibitor. You need something much more sophisticated. What else?
Second, primate data is absolutely essential, non-negotiable. The way mutations accumulate, the efficiency of repair, the whole genomic stability landscape, it's vastly different between a mouse that lives 2 years and a primate that lives for decades.
What works in a mouse brain over months might do nothing detectable in a monkey over 5 or 10 years.
Precisely. You need to show it works in a system much closer to humans before you can get excited.
And third, going back to that Alzheimer's correlation, you mentioned confounding factors.
Yeah, that finding lower DREAM predicts later AD onset. It's intriguing on the surface. But we have to be super careful about what I call healthy aging bias. Meaning people who reach old age in generally better shape, lower inflammation, better metabolic health, stronger systems overall, they might just naturally have lower DREAM activity as part of that healthy profile.
So DREAM isn't the target, it's just another marker reflecting overall health.
It could be. In that case, it's a great biomarker of robustness, but not necessarily the thing you want to drug. This feels like very early work that could easily, as we said, regress to the mean once you apply stricter controls in human studies. Needs much more validation.
Okay, let's shift gears then. Mechanism 2. Moving from the DNA sequence itself to epigenetic stability. This paper looked at the fundamental building block of chromatin, the nucleosome. It's titled Nucleosome Stability, Safeguards Cell Identity, Stress Resilience, and Healthy Aging, from Tanaka H et al. At Sanford Burnham Prebys, MDI, also on the bioRxiv, September 18th.
Right, this one takes us deep into the physics of the genome. We know aging messes with chromatin structure. Generally, things open up, things close down inappropriately.
Yeah, the epigenetic landscape changes.
But this paper asks a more basic, almost mechanical question. Does the inherent physical stability of that core unit, the DNA wrapped around the histone octamer, actually matter on its own, separate from the chemical tags, the methylation, the acetylation?
So just how tightly packed and stable that little spool of DNA and protein is, they're arguing that physical property is a conserved safeguard.
Exactly. A safeguard for maintaining cell identity, preventing that transcriptional chaos or drift we often see in aged or senescent cells. The idea is that a wobbly nucleosome makes it easier for the wrong genes to get turned on or off.
How did they test that? You can't just easily make nucleosomes wobbly in a living cell, can you?
Well, their method was clever, but also highly artificial. That's the key caveat here. They engineered artificial histone mutants.
Ah, okay.
Yeah.
Not naturally occurring variants.
No. These were specifically designed mutations intended to weaken the physical bonds between the histone proteins themselves within the octamer core. Basically, they deliberately built a structurally fragile filing cabinet for the DNA.
And what happened when they put these fragile cabinets into cells?
This is where it gets interesting. They saw a crucial distinction, short term. Things like overall chromatin accessibility, global DNA damage levels, even basic cell viability, they weren't really affected much.
So the instability wasn't immediately toxic or catastrophic?
Right. It suggests the cell can tolerate some level of this initially, but the long-term consequences for information fidelity, those were profound.
How so?
The induced instability significantly messed up lineage-specific gene expression programs. Cells started losing their clear identity. You know, a liver cell needs to act like a liver cell. These cells started getting confused transcriptionally.
That sounds very much like a hallmark of cellular aging. Loss of identity, impaired function.
It's a core feature. And furthermore, this loss of identity wasn't silent. It directly triggered intrinsic inflammatory pathways and generalized stress responses within the cells.
Which connects it back to known aging phenotypes, right?
Mm.
That link between genomic or epigenomic instability and inflammation is pretty well established.
It is. And seeing it triggered here by purely physical instability is suggestive. It implies the structural breakdown itself might be a primary signal for these stress pathways.
Did they show this was conserved? Did it happen in other organisms?
They did a good job there. They validated it across a pretty wide range from yeast, Saccharomyces cerevisiae, up through worms, C. elegans, and flies, Drosophila, and in human cells too. In all these systems, forcing this nucleosome instability led to faster age-related decline or lower resilience. Suggests it's a pretty fundamental mechanism.
Okay, so critically evaluating this one, the novelty seems high. Focusing on physical structure is a bit different.
Definitely a novel angle within chromatin biology.
But, as you flagged, the whole thing relies on these artificial histone mutants. How on earth do you bridge that gap to something therapeutically relevant? That seems like a massive leap.
The gap is enormous, yeah, because in real aging, nucleosomes probably don't become unstable because of some sudden widespread mutation in the core histone proteins themselves. That's just not plausible.
So what would cause instability naturally?
It's much more likely due to things like problems with histone chaperones, the proteins that help assemble and disassemble nucleosomes, or maybe certain post-translational modifications affecting stability, or even metabolic changes altering the availability of, say, acetyl-CoA needed for histone acetylation, which can impact structure.
So the artificial mutant is a great proof of concept tool, but it doesn't point to a natural target we can easily drug.
Precisely. It proves the principle stability matters. But it doesn't tell us how stability is lost in normal aging, or how we might restore it pharmacologically.
And even if we knew the natural cause, is stabilizing a protein complex like the nucleosome even feasible with a drug?
That's the other huge hurdle. Trying to stabilize a protein-protein interface, especially one as fundamental and conserved as the histone octamer, it's way harder than, say, inhibiting an enzyme's active site.
Why is it so much harder?
You'd need a small molecule that can get into the cell, into the nucleus, find the specific interface between histone proteins, bind there, and somehow physically strengthen that bond. All without gumming up the works, causing chromatin to become too compacted globally, which could silence essential genes.
Sounds like a nightmare for medicinal chemists.
It's a monumental structural biology and drug design challenge. Not impossible, perhaps, but incredibly difficult.
And one more point, the downstream inflammation. You noted that instability triggered inflammation, which looks like aging, but couldn't that just be a generic stress response?
Mm.
If you mess with the genome or epigenome enough, doesn't it almost always trigger inflammation?
That's a very fair critique. We need the mechanistic link. Does the physical instability, for example, expose bits of DNA that shouldn't be exposed, maybe cryptic promoters or transposable elements?
And the cell sees that as foreign or damaged, triggering an immune response.
Exactly, like NF-kappa B activation. If researchers could show that stabilizing the nucleosome specifically prevents that aberrant exposure and then prevents the inflammation, then you have a clear, potentially actionable pathway. Until then.
It's valuable fundamental biology, confirms physical integrity is important, but miles away from a longevity drug.
That sums it up well. Foundational work, not yet translational.
Okay, mechanism 3 takes us into arguably more speculative territory. Macro-level ideas about lifespan limits, the epigenetic clock and maximum lifespan. This is the paper, Estimating Universal Mammalian Lifespan via Age-Associated Epigenetic Entropy by Alba Linares, J.J. et al. From CNN CCLC, posted September 17th on the bioRxiv.
Ah, yes. This one definitely generated some discussion. It's provocative because it takes a concept we know works pretty well, the DNA methylation clock, pioneered by Horvath and others.
Predicting chronological age from methylation patterns.
Right. And it tries to extrapolate that concept quite aggressively, I'd say. They're moving beyond just predicting how old an individual is to proposing there's some kind of universal constraint on how long any mammal can live based on how fast epigenetic information decays. They call it entropy.
Epigenetic entropy. So the inevitable loss of information fidelity in the methylation patterns over time.
That's the core idea. Their method focused on quantifying this noise or randomness. They looked specifically at CPG sites that are evolutionarily conserved across many mammals, trying to find some fundamental rate of information decay common to all of us furry creatures.
How did they quantify entropy in this context?
They essentially use Shannon entropy, a measure from information theory that quantifies randomness or unpredictability. Applied here, it measures how predictable the methylation state, methylated or unmethylated, is at these conserved sites.
Okay, and the key finding?
The key finding was that the longer-lived species, whales, humans, naked mole rats, accumulate less of this methylation noise over time. Fewer of their conserved CPG sites become unpredictable or random as they age.
So, longer-lived animals are better at maintaining a low-entropy, predictable epigenetic state.
That's the observation. They seem to preserve that information fidelity better. And this led directly to their big headline-grabbing estimate.
The 220 years.
Exactly. They found that the rate at which this epigenetic entropy increases seems to decline in a surprisingly linear fashion as you look at species with longer maximum lifespans.
So faster entropy gain in mice, slower in humans, even slower in whales.
Correct. And if you just draw that line out, extrapolate it to where the rate of entropy gain theoretically hits zero.
You land somewhere around 220 years.
Roughly, yeah. Which they propose might be a universal maximum limit for natural mammalian longevity, dictated by this fundamental rate of epigenetic information loss.
It's a spectacular number. Great for grabbing attention, but you sound skeptical about the universal constraint part.
Deeply skeptical, yes. Look, a linear correlation across almost 100 species looks compelling on a chart.
It does.
But think about how differently mammals evolved. Shrews versus whales, bats versus elephants. We're talking vastly different metabolic rates, body sizes, reproductive strategies, environmental pressures.
Huge differences.
To assume that a single molecular process, the fidelity of DNA methylation, imposes a strict, universal linear constraint across that entire spectrum, it feels like a massive oversimplification, statistically and biologically.
Are they correcting for things like body size or phylogeny? Those are known lifespan correlates.
They likely attempt some corrections, but whether they can fully account for all the evolutionary divergence and differing selective pressures is highly questionable. The correlation might just be tracking something else we already know influences lifespan, like metabolic rate, which also scales with body size.
Okay, beyond the universality claim, let's focus on the concept of entropy itself. You hinted earlier, is this epigenetic noise the cause of aging, or is it just a really good readout or reflection of damage accumulating elsewhere?
That is the absolute crux of the issue for intervention. Is epigenetic entropy, this slow decay of methylation patterns, is just the downstream consequence of, say, accumulating DNA mutations, or mitochondrial dysfunction throwing off methylation enzymes, or chronic inflammation messing things up?
Then fixing the entropy directly won't fix the root cause.
Exactly. It would be like, constantly wiping away the dust without ever fixing the leaky roof that's causing the dust. Entropy is basically the second law of thermodynamics applied to biological information systems tend towards disorder. Is it an independent driver of aging or just the most visible manifestation of the system breaking down? We need proof it's a driver.
So what experiments would provide that proof? What does biotech need to see before taking reducing epigenetic entropy seriously as a strategy?
Two critical things, I think. First, intervention data from known lifespan extenders. Take something robust, like caloric restriction or rapamycin. Administer those treatments long-term in a model organism. Does it just slow down the epigenetic clock's age prediction, or does it actually demonstrably reduce the rate of entropy accumulation? Does it flatten the slope of that information decay curve over time?
Because if those interventions extend lifespan without changing the rate of entropy gain,
Then the entropy metric is decoupled from the causal mechanism of lifespan extension. It's a good marker, maybe, but not the target itself.
makes sense. And the second critical experiment.
The flip side, can we directly intervene to reduce epigenetic entropy and see a corresponding increase in lifespan? For instance, maybe through carefully controlled, periodic, partial epigenetic reprogramming, pushing cells back towards a younger methylation state?
If actively lowering entropy causes the animal to live longer and healthier.
Then the theory holds water. Then maybe that 220-year estimate represents a real, albeit perhaps modifiable, biological constraint. But until we have that causal evidence...
It remains a fascinating theoretical calculation based on correlation, a great conversation starter, but not something you'd base a clinical trial on.
Precisely. Intriguing science needs experimental validation to become actionable.
All right, let's ground ourselves again with mechanism 4, looking at actual human data, metabolomics, and extreme human longevity. This paper draws on that fantastic resource, the New England Centenarian Study, NACS. It's Metabolomic Signatures of Extreme Old Age by Montai, S. et al. From Boston University, posted September 16th.
Yeah, the NECS is invaluable. Studying people who've successfully navigated a century or more, they're the living proof of principle for longevity. They've somehow optimized their biology against all the usual slings and arrows.
And this study went broad, using untargeted serum metabolomics, looking at over 1,400 different metabolites.
That's right. In 213 NECS participants, the centenarians themselves, their offspring, and controls, they were hunting for the metabolic fingerprint of successful extreme aging.
Did they find one? A distinct profile.
They did report a distinct profile in the extreme longevity EL group. It seemed to be characterized by having persistently higher levels of certain molecules compared to the controls.
Higher levels of what, specifically?
The most striking were primary and secondary bile acids. They specifically called out chenodeoxycholic acid, CDCA, and lithocholic acid, LCA, as being elevated.
Bile acids. Interesting. What else?
Also, higher levels of biliverdin and bilirubin pigments related to heme breakdown, which might suggest really efficient liver function and handling of oxidative products, and alongside that, generally stable levels of certain steroids.
And crucially, was this just descriptive, or do these levels correlate with outcomes?
That's key. They found that higher levels of these bile acids and some steroids were actually associated with a lower mortality risk within the centenarian cohort itself. So it suggests these aren't just passive markers of being old, they might actually be protective.
Okay, that's more compelling. Did they look at pathways? What might this metabolic signature mean functionally?
They did pathway analysis, yeah. They found inverse associations, meaning things linked to survival were inversely correlated with metabolite ratios indicating stress or inefficiency in certain pathways.
Like what?
Like potentially better NAD plus production efficiency based on tryptophan and urinane ratios, maybe optimized gut bacterial metabolism, looking at ergothionein TMAO ratios, and potentially lower oxidative stress burden based on methionine and methionine sulfoxide ratios. It paints a picture of a metabolic engine that's just running cleaner, more efficiently, less burdened in extreme old age.
They also mentioned developing a new metabolomic clock from this data.
Yes, based on these centenarian signatures, they claimed it identified key metabolites like taurine and citrate that were predictive of biological aging in their cohort but weren't really captured by standard age analyses. Potential new biomarkers of resilience, perhaps.
It sounds like a really rich data set. But what are the limitations here? Centenarian studies always have caveats, right?
The massive one is survivor bias. It's inherent to studying extreme outliers. These individuals are, by definition, the ones who didn't succumb to age-related diseases earlier. Is their metabolic profile the cause of their longevity, or is it simply the result of having survived everything else?
The chicken and egg problem. Does the metabolism make them live long, Or do you only achieve that metabolism if you live long?
Exactly. We can't easily disentangle that from purely observational data on survivors. And sample sizes, while decent for centenarian studies, are still relatively small when looking for robust signals among such extreme outliers.
Let's drill down on the bile acid finding, CDCA and LCA. You said that connects to the gut microbiome and liver function, which are already hot areas in aging research.
Right. Bile acids aren't just for digesting fats. They're crucial signaling molecules. They interact with receptors like FXR in the liver and gut and TGR5 on cell membranes, regulating glucose metabolism, lipid handling, inflammation, energy balance.
So elevated levels in centenarians might mean these protective signaling pathways are just cranked up higher or working better.
That's the hypothesis, that these individuals have optimized gut-liver axis signaling, potentially mediated by specific bile acid profiles, contributing to their metabolic health and longevity.
Okay, if they're potentially protective, the obvious question is, can we just supplement with CDCA or LCA? Give people more of these good bile acids.
Ah, if only it were that simple. That's the major translational hurdle. Playing with systemic bile acid levels is tricky business.
Why? Toxicity.
Yes, bile acids themselves, especially certain types and at high concentrations, can be toxic to liver cells, hepatotoxic. They can disrupt bile flow, leading to cholestasis. There's a reason the body regulates their levels so tightly. Finding a safe and effective therapeutic window for supplementation would be extremely challenging. Are we aiming for systemic levels or just modulating gut signaling? Big questions.
So just because it's high in centenarians doesn't mean giving more to everyone else is safe or effective.
Not at all. It's a classic case where the dose makes the poison.
Right.
We first need to understand, do these protective metabolic patterns emerge early in life and those destined for longevity? Is it a lifelong advantage? Or is it a late-life adaptation?
And the critical experiment.
Can you take a normal model organism, give it safe, moderate supplementation with something like CDCA or LCA, and actually extend its health span or lifespan? Especially in an organism that doesn't already have the genetic background of a centenarian.
You need that causal evidence in a non-elite system.
Exactly. Without that proof, the centenarian metabolome remains a fascinating, invaluable map of what biological success looks like, but not necessarily a direct recipe for achieving it pharmacologically.
Okay, let's pivot now for our final mechanism, Mechanism 5, computational methods. We're shifting focus from the biological targets themselves to the tools we use to measure biological age. This paper is Guided Multi-Agent AI Invents Highly Accurate, Uncertainty-Aware Transcriptomic Aging Clocks. Agarwal, V. et al. From MIT, Biostate.ai, and Baishidi AI, posted September 12th.
Right, this is a really important area. As the field invests billions trying to develop longevity interventions, we absolutely need better rulers. Rulers that are not just accurate, but also tell us how confident we should be in their measurements.
And this paper claims an advancement on that front, particularly regarding confidence or uncertainty.
That's the main thrust, yes. They developed this AI system called Cadence, trained it on a huge transcriptomics dataset, RCHS4, over 57,000 samples, 28 tissues. Massive amount of data.
And the performance. How accurate was the clock they built?
The numbers are good for a pan-tissue clock based on gene expression. They reported an R-squared of 0.854 and a mean absolute error, MAE, of 4.26 years for their main unified ensemble clock.
R-squared 0.85, MAE around 4 years. That sounds pretty solid.
It is solid, but let's be clear, those numbers represent a good but likely incremental improvement in raw accuracy compared to other state-of-the-art transcriptomic clocks already out there.
The real novelty isn't just shaving a bit more off the error.
Exactly. The core advancement, the thing that makes this paper stand out methodologically, is building calibrated confidence intervals right into the prediction. It doesn't just give you an age estimate, it tells you how certain the AI is about that estimate.
Uncertainty quantification. Why is that so critical for, say, clinical trials or practical use? If a clock is already highly accurate on average, does the uncertainty band add that much?
It adds a huge amount, especially when dealing with real-world biological variability. People are incredibly diverse, biologically speaking. That diversity increases at the extremes of age, very young or very old, and also during major life transitions like puberty or during illness.
Okay.
An aging clock might give a precise number, but if the underlying biology of that individual at that moment is highly unusual or unstable, the prediction could be way off. This AI, by providing a confidence interval, essentially flags those cases, says, here's the estimate, but beware, I'm not very confident about this one.
So for a clinical trial, you could potentially filter out or downweight predictions where the AI itself signals high uncertainty.
Precisely. You could avoid being misled by predictions based on transient biological noise or extreme underlying conditions not related to the intervention being tested. It improves the signal-to-noise ratio of your results. A prediction that knows its own limitations is far more valuable than one that's just precise but sometimes wildly wrong.
The AI approach also seemed to speed up discovery, identifying markers relevant at different life stages. They mentioned things like CDKN2, AP16 for senescence, septin-N3 for neurodegeneration resistance.
Yes, that's another strength of these guided AI approaches. The multi-agent system can explore the vast feature space of the transcriptome much faster than humans can, compressing potentially months of analysis into weeks. And their sliding window analysis was neat, too.
What did that show?
It showed that the genes most important for predicting age actually change over the lifespan. It's not a static set of markers. They described these wave-like shifts, meaning the transcriptomic signature of aging is constantly evolving. That challenges simpler models that rely on a fixed set of genes.
Okay, impressive computational work. But let's apply the skepticism again. It's primarily a methods paper developing a better tool. How do we ensure this more sophisticated ruler is actually measuring something meaningful for longevity interventions?
That is the million-dollar question, isn't it? Or maybe billion-dollar question for the industry. Just being highly accurate at predicting chronological age isn't the ultimate goal.
Right, a calendar does that perfectly.
Exactly. The real value of a biological aging clock lies in its ability to predict functional decline, disease risk, and most importantly for biotech, response to interventions. We've had accurate clocks before that didn't reliably predict who would benefit from, say, metformin or rapamycin.
So the critical test for this cadence clock, or any new clock, isn't just its R squared against chronological age.
Absolutely not. The crucial validation step that's often missing is, does this highly accurate, uncertainty-aware clock predict the success or failure of specific anti-aging drugs or lifestyle changes better and earlier than existing clocks?
Can it tell you within, say, 6 months of starting a trial whether the intervention is likely moving the needle on long-term health span or lifespan?
That's the benchmark. If Cadence, with its uncertainty awareness, can do that more reliably than previous tools, then it's a genuine breakthrough for drug development. If it just gives a slightly more precise estimate of chronological age in untreated populations.
Then it's just a fancier calculator for a metric whose ultimate relevance for intervention is still being debated.
Precisely. Its true value proposition lies in predicting response and trajectory changes, not just nailing the baseline age estimate.
Okay, so let's try to synthesize what we've seen this week. Looking across these papers, there's definitely a pattern. It feels like high-quality work, but very much incremental progress across the different hallmarks.
I agree.
We saw refinements in understanding DNA repair regulation with the dream complex. We got a deeper look at epigenetic stability via nucleosome physics. We saw efforts to measure epigenetics better, both through this macroentropy concept and the practical AI clock. And we mapped some interesting metabolic details in centenarians.
The overall picture reinforces that opening sentiment, doesn't it? It's the 95% probability scenario playing out. The field is getting incredibly good at collecting complex, high-dimensional data. We're drawing clearer lines connecting fundamental biology like dream activity influencing mutation rates to lifespan, at least in models or via correlation. And the tools, like that AI clock, are getting impressively sophisticated.
Right.
But the leap remains profound. The jump from finding a protective signature, like those bile acids in the NECS cohort, or observing a theoretical limit based on entropy, to actually designing an intervention that's safe, effective, and truly shifts the paradigm, something that reliably adds, say, 10+ healthy years to human life. That's still firmly in the experimental realm.
We're still translating the descriptive language of successful aging biology into the prescriptive, engineered language of medicine and pharmacology.
Well put. And maintaining that critical perspective is essential. Every paper we discussed adds a piece to the puzzle, clarifies a part of the map, but none of them handed over the keys to the longevity kingdom this week.
Definitely not. Okay, let's wrap up with a final provocative thought for you, our listeners. Let's try to connect two of the ideas we wrestled with today.
All right, let's try it. Consider the tension between these two papers, the theoretical versus the mechanical.
Okay.
On one hand, you have the Alba Lanner's paper, proposing this potential universal lifespan limit of around 220 years, based on the seemingly inevitable decay rate of epigenetic information, that entropy concept.
Right, the fundamental information loss limit.
And on the other hand, you have the Koch paper on the dream complex, suggesting we can mechanically intervene, tweak the DNA repair machinery, and demonstrably reduce the rate of somatic mutation accumulation, at least in mice.
So a potential hard limit versus a potential mechanical fix. Where's the tension?
The question is, how do these interact? If that 220-year epigenetic entropy limit is real and fundamental, does it mean that even our best efforts to perfect DNA repair by inhibiting DREAM are ultimately capped? Will the software epigenome inevitably degrade at a certain rate no matter how well we preserve the hardware DNA sequence?
Is the fidelity of the epigenetic operating system the ultimate bottleneck, regardless of fixing bugs in the underlying code?
Or could it be the other way around? If we get really good at interventions like DREAM modulation and we successfully slow down somatic mutation accumulation over decades, will that preserved genomic integrity in turn help maintain the fidelity of the epigenetic information? Could it actually flatten that epigenetic entropy curve?
Thereby potentially pushing past that predicted 220-year limit.
Exactly. Could fixing the hardware fundamentally stabilize the software? Figuring out the true causal relationship between sequence fidelity and epigenetic fidelity over the long haul? The answer to that might really tell us whether engineering human lifespan significantly beyond its current natural boundaries is truly feasible.
A fascinating interplay to consider. Something to definitely mull over. Thank you for joining us for this analysis of the longevity papers.
Always a pleasure.
We absolutely encourage you to keep digging into the data yourselves, always with that intense critical lens. See you next time.